Hello everyone, myself Professor S.K. Sonone from Kekewa College of Agriculture, Nasik, Department of Plant Pathology. Today we discuss today's presentation on bacteriophage. Bacteriophage. This is the diagram of bacteriophage. Bacteriophage is any one of a number of viruses that infect bacteria. The simple definition of bacteriophage is virus infecting bacteria. It is called as bacteriophage. They do this by injecting genetic material which they carry enclosed in an outer protein capsid. This is the process. Bacteriophage process takes place by injecting the genetic material into the outer protein capsid. The genetic material can be single-stranded RNA, means ssRNA, or dsRNA, double-stranded RNA, ssDNA or dsDNA, single-stranded DNA or double-stranded DNA. SS or DS prefix denotes the single strand and double strand, along with either circular or linear arrangement. French-Canadian microbiologist, the scientist French-Canadian microbiologist Félix de Herelle, working at the Pasteur Institute in Paris, discovered an invisible antagonistic microbe of the dysentery bacillus. De Herelle called the virus bacteriophage or bacteria eater from the Greek phagein, meaning to eat. He also recorded a dramatic account of a man suffering from dysentery who was restored to good health by the bacteriophage. Replication of lytic cycle. In bacteriophage, two cycles takes place, that is lytic cycle or lysogenic cycle. The bacteriophage may have a lytic cycle or lysogenic cycle, and a few viruses are capable of carrying out both. With lytic phase, such as the T4 phages, bacterial cells are broken open, lyse, and destroy after immediate replication of the virion. As soon as the cell is destroyed, the phage progeny can find new host 10 infected. Lytic phages are most suitable for phage therapy. Some lytic phage undergo a phenomenon known as lysis inhibition, where completed phage progeny will not immediately lyse out the cell if there is high extracellular phage concentrations. This mechanism is not identical to that of temperate phase going dormant and is usually temporary. This is the diagram of lytic cycle. First step, host cell attachment, then penetration into the host cell, virus penetration into the host cell, then step 3, biosynthesis. Then in step number 4, assembly, virus are attached in a assembly position, and then release, step number 5, release from the bacterial cell. Then lysogenic cycle. The lysogenic cycle does not result in immediately sign of the host cell. Those phages able to undergo lysogeny are known as temperate phages. Their viral genome will integrate with host DNA and replicate along with it fairly harmlessly, or may even become established as a plasmid. The virus remains dormant until host conditions deteriorate, perhaps due to depiction of nutrients. Then the endogenous phages known as prophage become active. At this point, they initiate the reproductive cycle, resulting in lysis of the host cell. As the lysogenic cycle allows the host cell to continue the survival and reproduce, the venus is reproduced in all the cells of spring. Sometimes prophage may provide benefit to the host bacterium while they are dormant by adding new functions to the bacterial genome in a phenomenon called lysogenic conversion. It's called as lysogenic conversion. An eminent example of the conversion of harmless strain of Vibrio cholera by a phage into highly virulent one which cause cholera. Means the lysogenic cycle takes place in Vibrio cholera. This is the cycle of difference between the lytic cycle and lysogenic cycle. Okay, we already see the lytic cycle. The virus penetrate into the bacterial cell, then assemble and then replicate and then release out of bacterial cell. In the lysogenic cycle, virus grow with the bacterial cell nucleus and then replicate and then release from one place to another place. Thank you very much for listening this important topic of your theory syllabus, bacterial cell, bacteriophage.